The next presentation is entitled Modeling for Ambiguous S or SNP cells in Allotetraploid peanut. Roshan Kulkarni is our speaker.
Hello, everyone. I'm Roshan, and I'm from Iowa State University. Today, I'm going to talk about the most commonly asked question in last few years is how to identify variation in allotetraploid peanut. So peanut genome, as you all know, is allotetraploid. That means it has a combination of A and B genome merged in a single genome. And because of all these complexities, in general, peanut is a difficult crop to genotype. In addition, peanut is an inbreeding crop, and it is known to have a low genetic variation. So thus, we need a tool which can accurately identify SNPs and also does not filter away the useful information. The commonly used genotyping technique is a sequence-based, and it mostly involves identification of SNPs, that is single nucleotide polymorphism. When it comes to SNP, in allotetraploids, there can be different scenarios. So the blue segment here represents the A genome of the two genotypes, and the yellow segments are the B genomes for the two genotypes. In the left, you can see for genotype 1, there's a homozygous TT call in the A genome, and there's a homozygous CC call in A genome of genotype 2. So this SNP is actually differentiating the two genotypes between the A genome. Similar is the case below. This SNP is differentiating two genotypes in the B genome. However, on the right, you can see a heterozygous TC call in both subgenomes for the two genotypes. The difference between this SNP and towards the left is that this SNP is differentiating a single genotype within the subgenome itself, rather than differentiating the two different genotypes. Now, these distinctions are very clear when there is a proper assignment of the read after the genome alignment. But the challenges are the most commonly used pipelines to identify SNPs are traditionally designed for diploid organisms. And proper assignment of genotype reads to subgenome is not trivial because of the high similarity between the subgenomes. And they fail to identify these type of distinctions in allotetraploids. So there has been previous efforts to genotype allotetraploids. Tools like Sweep, HaploSweep, SweepML, all developed by University of Georgia. And there have been reports of GATK-based filtering methods that have been effective in calling SNPs in allotetraploids. But this is a complex problem, and there's a room for improvement. And every method has its advantages and disadvantages. So the idea is if you have a tool with a different methodology, you can use all of them together to make a proper, accurate genotype calling. So below is a process, is a general process used in SNP calling when you do genotype by sequencing. You do basically the first step is the alignment of your reads with the reference sequence, and then you call SNPs using different SNP calling methods. As you can see, the other methods actually work much more downstream in the pipeline. And our proposed method work in the first step itself. So this is one of the most different methodological scheme than other previously reported methods. I'm just going to go over some of the terminologies and some of the color schemes which I'll talk in next slides. So I have a genome reference segment and a B genome reference segment represented in blue. And nucleotide represented in pink is the difference at the same site in A genome and B genome reference sequence. So this pink nucleotide, I will call it as homoeologous SNP. which is differentiating the reference genome itself. When you look at two genotypes and two respective genomes, if there is a variation that is differentiating two different genotypes within the respective subgenomes, I call it homologous SNP, and I color them differently, representing whether it is coming from A genome or B genome. And of course, if they are not segregating, they're homozygous, and if they're segregating, they're heterozygous. And in addition, because of presence of repeat sequences in peanut genome, there can be paralogous sequences and SNPs resulting from those sequences. So this is an overview of our approach. There are two main schemes to this. One is the data derived from amplicon sequencing, and the second is data derived from whole genome sequencing. We treat these both differently because they have a different kind of problems to deal with. When you consider amplicon sequencing, you have high read depth, and because of that, which is great, but it also comes with consequence of a lot of paralog sequences being captured and a lot of PCR artifacts and so on. So we do paralogous filtering before genotyping in this case. For whole genome sequencing, since the data is huge, in order to save time, we subset these sequences based on the region of interest prior to genotyping. Let's talk about amplicon sequencing first. So this is a general approach. The first step is paralog filtering. So we align the Illumina reads, raw Illumina reads, to both A and B genome. Here I represent the homoeologous SNP as mentioned before, and there is a variation in A genome, and this is a variation, known variation in the B genome. So when you're aligning the amplicon sequences, since there's presence of paralogs, these paralogs with unintended or unknown SNPs also align to these reads. And our goal is to filter this first. So we pick the first step is to pick the top foremost abundant groups. We do that using a tool called AmpliCI. We do top foremost because at most we can have two unique sequences for A genome and two unique sequences for B genome. And considering that the paralogous sequences are the result of errors and the read depth will be low, that if we capture the top foremost, we should get only the legitimate reads that are not paralogous. But there can be a case where both the variations are not present. There can be a case where, for example, there are two unique sequences from A genome, but there's only one unique from B, or the other way. Or there can be just one unique sequences that is it is homozygous for A genome and homozygous for B genome. In that case, when you pick the top foremost, you're actually picking the third, or in this case, the last two paralogous sequences. So we need to consider all these cases in order to clearly filter these paralogous sequences. In this case, as this site is heterozygous and this site is heterozygous, this is case one, wherein you have two unique sequences for A and B, and our top foremost groups will be devoid of paralog sequences. After this, we start the process of genotyping. So we do side-by-side genotyping, that is, we genotype each basis, and then we also consider one read at a time. This is one more variation from the other methods that we can genotype a single individual. We don't need a group of individuals in order to genotype an individual. So for this slide, let's consider a site which is homologous SNP. So the first thing we do is find the most common allele for that site and second most common allele for the site, and we call it major and minor allele. Of course, there are I reads here, but if you just consider 4 reads, 4 in B, 4 in A, and 4 in B, that is in total 8. you can see there are 4 A's and 4 C's, so it's kind of a tie. So our major allele would be A and minor allele would be C in this case. And then we look at the differences for each reads against the references. So if you consider read 1, there is actually virtually no difference between this read and the reference sequence. But if you consider this read aligned to B genome, you see 3 different differences in 3 bases. So the probability of this being true for alignment against A would be much higher than this because of these differences. So we then do genotyping based on the highest posterior probability for all allele combinations. So for this site, we check for all different allele combinations. So the variation here is AC. So we check for all different types. So AA is for the A genome, and this part after the slash is for B genome. So these are the A genome alleles and B genome alleles. So we check for all different possibilities. In this case, the highest probability for AA will be for AACC. So the genotype for this site for A genome will be AA, and for B genome will be CC. So now coming to the whole genome sequence genotyping. So as peanut genome is huge, and probably if you would have realized by now, if we go for side by side and read by read, it will take a lot of time to genotype the regions. Most often in plant breeding, we don't need to genotype whole 3.2 billion base pairs. We already have a lot of information from previous studies about some of the interesting regions. So user can actually select a certain region window. So the first step in genotyping whole genome is we align the reads to whole reference A genome and B genome, but then we subset the alignments based on the selected target region. So now you're basically genotyping just few thousand base pairs compared against the 3.2 billion base pairs. So once you break it into this eventually becomes a case like genotyping amplicon sequencing, which I mentioned before. Apart from 2 notable differences, if you would have noted my previous slide, all the reads, the start site and the end site were same. That's because it was amplicon sequencing. Now, since we're breaking it in the middle of the whole genome, the start and end sites are different. So that's the modification in the code when we deal with whole genome. And also the read depth of when you have whole genome sequencing is really low. So you most often no need to do a paralogous filter. So for validation, so we had 2 cases, amplicon sequencing and whole genome. And we did do simulation for 2 reasons. One is because we know the absolute truth of what kind of SNP are presented at what position and to know what we are looking for. And then to show our applicability in real data. But for time constraint, I'm going to just summarize results for just one of the cases. So these are the results for amplicon sequencing for the simulation data. Red is the SNPs identified in the A genome. B is the SNPs identified in the B genome. And these 2 colors are the false positives identified in respective subgenomes. And I have 3 different categories of SNPs: homozygous or true SNPs, homoeologous, that is the SNPs between A and B genome, and that's why
we have 1 minute left for this presentation.
And then you have heterozygous SNPs. As you can see in the GATK tool, there are a lot of false positives and high number of heterozygous SNPs compared to our approach. And this is an example of the SNP identifier. So in summary, we have validated this on simulator and real data for amplicon and whole genome. And we are currently processing high depth data for whole genome. And we are working to see the limits of this tool with varying parameters. With that, I would like to thank my advisors, Dr. Stephen Can and Dr. Dorman, and my lab members, UD, Andrew, and Paul.
All right, thank you. I'm sorry we don't have time for questions, but thank you for being here with us.
All right, thank you.